And we have a big initiative that that we have started, and we regularly meet and try to develop programs such that we can address these issues. So we welcome you to join these efforts. This is the IQB DEIA committee. Professor Wei Dai is the chair, and there are several members from IQB faculty and staff. And so I invite you to engage with us and talk about these really important issues that we want to make a contribution to. And we welcome your contributions as well. A few announcements about upcoming events at the IQB. So first, on the 7th of December, we're going to have a virtual crash course. This is a day-long event where you get to listen to talks by some of world experts in a particular area. So this time, it's going to be about the important topic of cancer immunology and metabolism. And there is more information available, and you will need to register if you want to attend. It's open to everyone, but you need to register. So please go to this link and register or search for this on the IQB website. And then the second event that I want to draw your attention to is the 10th annual winter boot camp. So we have these boot camps every winter, sometimes in the summer as well. And this is a week-long event where all participants, which include undergraduate, graduate students, postdocs, and sometimes even faculty, will work closely with experts on writing and molecular visualization, and will write essentially a molecule of the month article that will be featured on the Worldwide Protein Data Bank website. And scholarships are available. And although application deadline has passed, and I'm told that all slots are filled, so unfortunately you won't be able to join unless something changes. But do get in touch if you want to join, and we might have a wait list. And so this is the IQB. This is our team. That's Stephen Burley, and and all of us are here trying to do and support science, as well as increase participation of everyone in science. So with that, let me stop sharing and introduce our speaker. So it is my great pleasure to introduce Anum Glasgow. Anum comes to us from Columbia University Medical Center, where she's an assistant professor, a newly minted assistant professor 11 months ago in the departments of biochemistry and molecular biophysics. Before Columbia, she was at UCSF working with Tanya Korteme, where she did some really beautiful work on both looking at Allosteric, but also in response to the pandemic, developed some really powerful receptor traps against SARS-CoV-2. Before going to UCSF, she was a grad student at Berkeley, I think, working on the type 3 secretion system, and was an undergraduate at Johns Hopkins before that. So today she's going to tell us about her work on decoding evolutionarily conserved allosteric signaling in a family of transcription factors. And I'm really excited because I think from the point of view of protein design, design of allosteric certainly remains one of the big challenges in the field. So I think the insights that Anum has obtained, I think will be really important. So looking forward to that. And without further ado, Anum, take it away.
Okay, thank you for the invitation, and thanks for coming to my talk. I'm really excited to be here. We are still figuring out how to do that kind of design, but today I want to tell you about the first half. What did I do in my postdoc to figure out the allosteric mechanism of one protein? And then in the second half, what we're working on now to try to extend that to, for example, protein design. Okay, so we would love to be able to design allosteric proteins because many natural proteins work like that, where you poke a protein in one place and you get a functional response somewhere else in the cell, in the protein, and then you can get a new functional protein that can go and do something in the cell. But there are many kinds of triggers for protein allostery, right? So you can have like a ligand binding interaction of a protein with a small molecule or peptide or protein. In my opinion, an underrated form of regulation is oligomerization, where you can expand the number of functional units in the cell by making it such that a monomer does one thing and a dimer does something else. And of course, proteins work by responding to these kinds of molecular triggers by changing their conformation. And of course, in solution, proteins occupy many different structures, but the number of functional state proteins might change in response to binding one or another thing or undergoing like a post-translational modification, etc. But it's really hard to watch proteins do these kinds of behaviors. So, we don't really know how to encode it de novo. So, for example, how do we engineer conformational changes in proteins where we don't have a structure, right? Like a lot of proteins will undergo a disorder to order transition, but if we don't know what the protein looks like in its disordered state, we have a hard time engineering it to undergo this type of transition. There's also the problem of combinatorial regulation, right? We might know what a protein looks like in its low energy state when it's bound to one thing or bound to another thing, but sometimes proteins bind two things, and then that is the functional state. It's hard to know how to do that when the structural transition is not well understood. Finally, there is the interesting problem of when there are no obvious changes in the average structure of a protein in response to a perturbation of some kind. Nevertheless, we want to be able to engineer the functional form of the protein. And so one such example is the lac repressor. You probably learned about it in biology, but I never took a biology class. It was really exciting for me to see the structure of this protein when I was halfway through my postdoc. The lac repressor looks like this. It's a paradigm transcription factor, and it's very well studied for understanding gene regulation. The protein is a dimer. It has a DNA binding domain, which it uses to bind to the lac operator. It also has a core domain. This is like the regulatory domain of the protein, which has two subunits, the N and the C terminal subdomain. We call it the core domain. And the lac repressor, it can bind many different ligands in the ligand binding pocket, which is right here, in between the two subdomains of the core, and this can change its functional state. For example, when an inducer molecule binds in this ligand binding pocket, then the DNA binding domain is understood to become less structured, so that the KD, or the binding affinity for the DNA operator, becomes higher. So, or the binding affinity is increased and the KD gets higher, so that polymerase can come and bind over there and gene transcription can occur. So, these ligand binding sites are in a really interesting region of the protein. This core domain has this like Venus flytrap architecture that is really well conserved, actually, and we find it again and again in proteins that are not just transcription factors, which I'm going to talk about a little bit towards the end of this presentation. But for the lac repressor, it can respond to a variety of ligands for different functional effects. So I mentioned inducer molecules. You may have used IPTG in the lab if you ever do protein expression in cells. It can also bind allolactose or TMG. These are all sugars that act as inducers. That way bind here, they destructure the DNA binding domain to get the gene expression. But there's another class of structurally and biochemically very similar sugars called anti-inducers that bind in the same ligand binding pocket, but they have the opposite functional effect and structural effect of increasing the affinity of the well-folded DNA binding domain for the operator sequence so that you get increased repression. We can see this in a simple cell assay like this, where we have the protein in cells chilling and it's producing some GFP under the control of a lac operator, but very little because it's repressed. When we add an inducer molecule, we get derepression and an increase in the production of our gene of interest. It's making lots of proteins, and then if we add an anti-inducer, we see an increase in the repression, decrease in gene expression. We can also measure in vitro the binding affinity for different DNAs. So here, if we increase the lac repressor concentration in vitro, in the absence of IPTG for different operator sequences, we can see the binding. And then in the presence of 100 micromolar IPTG in this experiment, we see a total loss of DNA binding. The really interesting thing about all of this is that actually all of these interesting functional effects can happen without large changes that we can observe in the structure of the ligand binding domain of the protein, or the core domain. So this is what the LAC repressor looks like. A couple of decades ago, there was a burst in the number of LAC repressor X-ray crystal structures. So we have like, a dozen of these, but they all look the same in the core domain of the protein. So here I'm showing an overlay of the apo, like non-ligand bound state of the protein, and also the inducer bound state, or IPTG bound state, and then also the ONPF, or anti-inducer bound state of the protein. And I hope you can appreciate that the global RMSD of the backbone of the proteins is very low. They basically overlap exactly. And then if you look closely in the ligand binding site, we can see that also the side chains occupy very similar positions. And so presumably these sugar molecules bind in this ligand binding pocket in a really similar way. So how do we get the signal transduction over this 40 angstrom distance between the ligand binding pocket and the DNA binding domain? So that's what we decided to figure out. First of all, how does a LAC repressor transmit this information from the core of the protein where the ligand binds to the DNA binding domain over this 40 angstrom distance with few observable structural changes in the core domain? And then why do we get different functional effects from these extremely structurally similar small molecules? So because X-ray crystallography didn't work out for this problem, and protein is a little bit small for cryo-EM, decided to try a technique called hydrogen deuterium exchange with mass spectrometry, which I learned at UC Berkeley in Susan Marcus's lab. It is a really cool technique. Let me briefly explain how does it work. So we have all of our different states of the protein. So we can prepare a LAC repressor in complex with inducer molecules, or with anti-inducer molecules, or with DNA, or even ternary complexes. And we take all of these proteins in solution, they're in aqueous buffers, and we dilute them into identical buffers, which instead of having hydrogens, they have heavy water, so they have deuterium. So over time, and the time course in our experiment is 4 hours, protein's undergoing thermal fluctuations in solution, and it naturally exchanges the hydrogens in the protein for deuterium in the buffer. And so what we can observe in this experiment is the exchange of the protein backbone amide hydrogens for deuterons in the buffer. Over the course of the experiment, we take aliquots of our protein samples, or protein ligand complex samples, and we quench the exchange reaction by putting them in an acidic solution and freezing them. And so this stops the hydrogen deuterium exchange. Then we proteolyze them by exposing them to pepsin or other proteases, acid proteases, and we mass spec the samples. And so what we get is a readout at different time points of the incorporation at each time point of the deuterium in the buffer for hydrogens that we started out with in the protein backbone. This gives us a high resolution picture of the conformational ensemble of the protein at these different time points. It's we get like peptide, in some cases, few amino acids at a time resolution because we proteolyze the sample. So we can say particular peptide in one state versus another, we had this much level of exchange or we had less exchange. And so we can interpret these kind of data to mean that, if you have more exchange in state 1 versus state 2, that means that it was in this region of the protein more flexible because it underwent more of these exchange reactions, because more of these hydrogen bonds that define the structure of the protein got broken. So anyways, for the LAC repressor, we can tune the conditions to get a pretty good coverage of our protein. The data actually look like this, right? So we have this mass spectrum for an IPTG bound LAC repressor for a particular peptide that has this sequence in water. And then at different time points, we can see a shift in the centroid of the isotopic mass envelope. And then we can fit these data to a nonlinear regression function. And we can do this for all 6 functional states of the protein for all of the time points and make these very simple uptake plots, right? So these uptake plots are useful because they can report on a lot of times grouping for different kinds of behavior for different regions of the protein. So here, what this peptide tells us is that in this region of the protein, we see some similar exchange behavior for the inducer-bound states of the protein as the apostate. We see something else, increased protection from exchange for the DNA-bound states, so presumably more rigid in those states, and then we see some in-between behavior for the anti-inducer in this peptide. And so we can expand this to the whole protein and get these kind of data. So I want to draw your attention to this particular helix, which is right at the, so this is one protomer, this is another protomer, and then the dimer interface is in between. I hope you can see my cursor because I'm pointing at a lot of stuff. Okay.
Yes,
so,
we can.
Okay, great. All right, so I'm going to talk about two different peptides in this helix. There's an N-terminal peptide here and a C-terminal peptide here. And we can look at, in the red, the DNA functional state behavior versus in blue, the IPTG bound state behavior. And we see really interesting effects for this helix, right? On the N-terminus in green now, we can see that when the protein is DNA bound, we see increased exchange and in the DNA bound state as compared to the IPTG state, and the opposite is true at the C-terminal part of this helix. In these experiments, I want to make it clear that we looked at the entire protein. Actually, we didn't, that's a lie. We looked at residue 1 to residue 330 of the protein. There is also a short tetramerization helix, which we removed because it's not necessary for the function of the protein. But we looked at the DNA binding domain and the core domain full-length dimeric protein. But I'm just going to show the core domain structure here because we are focusing on that because we have a pretty good idea of what does the DNA binding domain do, although I will return to that towards the end of the talk. But yeah, so this is the core domain. The N-terminal DNA binding domain would be down here. But for this helix, which is right at the dimer interface, we see that there is some difference in the IPTG bound state versus the DNA bound state from these different exchange patterns. We can expand this to the whole protein. I'm coloring it the same way as in the previous slide, where we can look at these different uptake plots and according to some stringency criteria, bin each part of each secondary structure in the protein according to its differential behavior in these two, like we can say physiologically relevant states of the protein, the inducer bound state versus the repressed state or DNA bound state. And so you can see like with some representative uptake plots on top here and on the bottom here, that like the teal parts of this protein are rigidified in the inducer bound state as compared to the DNA bound state. But all of these purple regions are like more flexible in the inducer bound state as compared to the DNA bound state. So even though the average structures of the protein look extremely similar, we can still see these structural effects from this cool experiment, HDXMS. Okay, so one of the reasons that I thought this was cool is because like the lac repressor has been extremely well studied and we have a lot of mutational data on the protein. So in the 90s, the Miller group at UCLA did a huge experiment where they made 4,000 individual point mutants of the repressor, which was a heroic effort at the time. And then they didn't look to see the individual biophysics of any particular mutant, but they looked for cell phenotypes, right? So then they binned each mutant into a phenotype category. And so a common thing that can happen when you mutate galactic broken response to the inducer molecule in this experiment IPTG. And so what that means is that either the protein no longer binds to the inducer molecule or something about the signal transduction is flawed so that you no longer get the communication from the ligand binding domain to the DNA. And so here I'm showing a structural representation of the core domain of the lac repressor with residues colored in blue that when you mutate them in this experiment, they saw this IS phenotype, insensitivity to IPTG. On the right, I'm showing the same structural representation of the core domain, but with residues colored in pink that have the effect of no longer allowing the protein to bind to DNA. And so in this case, you have like a constitutively on system, right? It's always transcribing the gene product because it can no longer bind to DNA at all when you mutate positions in pink here. And so what we saw was an enrichment in our set of positions from HDXMS data, right? For each of these kinds of phenotypes. So what I mean is that for these teal residues here, if we just look at these residues, 71% of these residues correlate they're at positions which are blue over here. So mutating them causes this insensitivity to IPTG. And then for the purple residues, we see an enrichment in this no longer responsive to DNA phenotype over here. You'll notice that there are a lot of residues in this other category, but it turns out most of these are quite mutationally tolerant from this original experiment in the 90s. After doing this initial classification, we sent the paper off for review, and one of the reviewers had a useful comment, I thought, which was like, what if you change the stringency criteria? So we did that, and then it turns out if you increase the stringency criteria for binning the HDXMS data in one category or another, actually this other category just decreases. And so we think that this effect is pretty real, actually, that like HDXMS data can be used as an indicator for some cellular phenotype level information. And so in our model, we think that even though the structures look really similar, they do represent different low energy states of the protein, which are defined by really like prototypical changes in the flexibility of particular secondary structures in the protein. And so these are important for the changed function of the protein. And so then you might ask, like, why do anti-inducers bind in the same position to have a totally different effect? And so looking at our HDXMS data for the anti-inducer bound state of the protein, ONPF, or the inducer bound state of the protein compared to the apo state where no ligand is bound, can shed some light on this phenomenon. And so here I'm coloring in green regions of the protein that are rigidified in response to binding individual ligands. On the left, the anti-inducer, and on the right, the inducer, that are rigidified in response to that binding event as compared to the apo state. And so what we can immediately see is that these patterns are different. And in the case of IPTG or inducer binding, we get the rigidification in unique positions local to the ligand binding site, but then in the ONPF case, we get some allosteric rigidification in like other parts of the protein more distal from the binding site. And so this was interesting to me because it's a pretty small ligand. So in crystal structures that we have of the IPTG bound state of the protein and in models that we can build of it, these don't actually make direct contacts with the ligand. So we hypothesized that maybe this is the effect of some solvent-mediated hydrogen bond network. That's tough to pin down experimentally. So we did some modeling of this, that maybe that there's like some important structural water molecules that contribute to the unique functional state of the IPTG bound state that allows for the induction. So I had a fantastic summer intern from the Rosetta RU program, Zion Perry, who spent her summer solvating computationally models that we made of the ligand bound states of the lactic repressor. And so here I'm cycling through 100 iterations of Zion's solvation protocol. At the time, this was a preprint, but now published, but useful method to find the positions of what we call structural water molecules. So these are water molecules that, in comparison to like transient molecules, make 3 or 4 hydrogen bonds with atoms in the protein or in the ligand. So we think that they make like an important contribution to the structure of the binding site in these different liganded states. If we zoom in on the ligand binding site in the IPTG bound state, you can see that we can compare the positions of these structural water molecules to crystallographic water molecules, and we get different classes of these. And so, for example, sometimes we get a water molecule predicted in a position always in the same place. Sometimes we get, a water molecule where the heavy atom is always in the same position, but the hydrogens change the positions depending on what they're interacting with in that model or in that simulation. And then sometimes also we just only sometimes get a water molecule at a particular position. And so we can consolidate all of these data in what we call water maps, which are pictures of the core domain of the protein, where side chains within 6 angstroms of the ligand binding pocket are colored according to the probability that they make hydrogen bond with a predicted structural water molecule using our approach. So on the left here, all of these like red and orange side chains in the ligand binding pocket make hydrogen bonds with structural water molecules that we predicted, like greater than maybe 90% of the time. Whereas you can see a comparatively sparser solvent-mediated hydrogen bond network in the ONPF model that we have of the protein. And so we compare this to our HDXMS data and see actually pretty good correlation with regions that are green here. So like rigidified upon inducer molecule binding and our water map, right? So, maybe this water molecules do contribute to the functional state structure. We see a similar correlation with the ONPF bound state, which gave me some appreciation for the like very apolar nature of this nitrophenyl substituent in the anti-inducer that we used here. So, we don't see any structural water molecules populated around this ring, but we see some around the hydroxyl groups in the fucoside. So we're excited about this because it's really difficult to pin down the role of structural water molecules in protein function. And it'd be great to do some like X-ray footprinting experiments to try to experimentally show that maybe these water molecules are important. We're trying to do MD simulations now to see if we can orthogonally corroborate some of these data, but nevertheless, we know that water molecules are really important in shifting the conformational ensemble of proteins. And so maybe here, this is an example of that. And so in our model for function in the lac repressor, in response to IPTG versus ONPF, we can start to make some claims about how the protein actually works. So here, this is like, a network diagram of the protein colored according to the differential rigidity in the inducer or anti-inducer bound states. And what we see in our HDXMS data and some of our simulations is that we have unique water-mediated and also just like ligand protein interactions that cross subdomains and also cross subunits in the IPTG bound state. So when IPTG binds, we see that more of these hydrogen bonds are possible. And we think that this leads to like a very small movement in the N-terminal subdomain of the core that destabilizes the hinge helix in the DNA binding domain, which binds in the minor groove, such that it leads to destabilization of the rest of the DNA binding domain. We think that this is how it works, and we can localize some of these effects to particular secondary structures. But by contrast, what we see in the ONPF bound state is that none of these structural changes that we observe are actually incompatible with the DNA binding. And in fact, because in the IPTG bound state, we see a lot of mutually exclusive effects on the structure of the protein as compared to the DNA bound state. But in the ONPF bound state, some of the largest differences that we see from other states are in actual structuring allosterically of loops in the N-terminal subdomain of the core, which could actually serve to further stabilize this part of the protein and even perhaps pre-organize the DNA binding domain. So I haven't talked much about this DNA binding domain, but I want to address it now. It was really hard initially to get good HDXMS data protein, and we thought, just like everybody else, it was because this part of the protein is destabilized in the absence of DNA, and maybe that's why nobody could ever solve the structure of it when there's no DNA around. But more recently, we've been able to get better quality data for this from these experiments. And so let me talk about that a little bit. And so what does the DNA binding domain look like? It's a classic helix loop helix domain. So lots of transcription factors and DNA binding proteins have it. So there are two helices that bind directly with the DNA operator, which is a symmetric palindrome sequence. And the two helices that make direct interactions with the major groove of the operator sequence interact both with the bases to make like stacking interactions, but also make electrostatic interactions with the phosphate groups on the backbone. Then there is a third helix that we think acts in a more supporting role, and finally the hinge helix that is then connected to the core domain of the protein by a short linker. And so in our initial experiments, we could see some effects in this domain. So these are wood plots. I'm showing 3 of them for 3 different time points in our experiments, and each line here is individually a peptide. And then on the y-axis, we see like level of deuteration, and then on the x-axis, it's a residue index. And so you can see from this top plot, let me erase this one for a second, sorry. We can see that in the DNA bound state, there is, especially at earlier time points, markedly decreased protection, or I should say exchange, in the DNA bound state as compared to the apo state or IPTG state. We see way less deuteration in these red lines as compared to these blue and green lines. It makes sense. We added DNA to the protein and nothing else, so it's DNA bound. So those parts of the protein we would expect to be much more structured. But we were kind of interested to see that like even these orange lines in this early time point at least also show increased protection from exchange, just like the DNA binding domain does. We didn't add any DNA to this experiment, we just added our anti-inducer. And so this supports the idea that perhaps this part of the protein is allosterically stabilized just by the addition of the anti-inducer molecule, such that the better binding kinetics for when you do add to the DNA, so as a mechanism for anti-induction. And so looking more carefully at this more recently, we wanted to see, like, does the sequence matter at all? Can we get like higher quality data for these peptides, both of those things? So here is the sequence of the natural LAC01 operator, which binds most tightly in E. coli to the LAC repressor. And so here I'm showing the sequence. We actually used two like tandem identical sequences for these experiments, double-stranded DNA oligos. And so in red here, you can see it's not actually a perfect palindrome. There are a couple of mismatches. And actually, we and others have observed that if you make it completely symmetric, it actually is a tighter binding to the DNA operator, which leads to the interesting question of like, actually, why did it evolve to be like kind of imperfect? But nevertheless, so we used the natural operator sequence. We also looked at a different palindromic sequence from E. coli, which naturally binds to the ribose repressor. So this is palindromic, but doesn't bind to, well, it hasn't been shown to bind to the LAC repressor. And then finally, we took the LAC operator sequence and just like scrambled it. So this is a scrambled operator sequence. And we look to see in the same exchange experiment, how well does it bind? And so here I'm coloring different regions of the DNA binding domain and showing representative small scale HDX-MS experiment. And so what we can see from these data is that for each of these peptides, the scrambled operator binds, maybe it doesn't bind that well, but it behaves like an apostate protein. But then when we add the natural LAC operator, we get increased protection from binding rather well. And then we have this intermediate state from the ribose repressor operator binding. So maybe even though we have a LAC repressor, not its cousin, the ribose repressor, perhaps it still recognizes the, some geometric parameter, I don't know, like something that is unique to a palindromic sequence in the protein and binds a little bit more tightly to that, because we see like the only region of the protein where we don't see a difference between the ribose repressor and the apostate and the scrambled operator as compared to the native LAC operator 1 sequence is right here, super far away from the DNA binding domain in the C-terminal subdomain dimer interface. And so here I'm showing this green peptide and this purple peptide here and here. And what we think is happening here is that there is some like molecular recognition effect that changes the monomer dimer equilibrium of the protein in its DNA bound state. So that one of the effects of binding really specifically to this particular sequence with high affinity is that you get everything in a dimer state, as compared to with non-operator sequences. Maybe it can't bend the DNA as much, whatever it is, it forces it into a different monomer dimer equilibrium. And so this is something that we are exploring a little bit now. Yeah, so some of our projects now are related to evolution and some are related to engineering. And I'm going to spend the last part of this talk discussing some of these new directions, although we don't really have anything like complete yet, but I'm excited about it. So in terms of evolution, we are really interested to see how many of these conformational changes that we discovered from these HDXMS experiments are really important for this effect to like release a transcription factor from DNA, right? So there are maybe 200 proteins in the LAC-I-GalR family. There's the LAC repressor, there's the ribose repressor, there are many other repressors. Also, some are activators. They share a conserved fold, but have less than 25% in most cases sequence identity. So it's a very ancient family of transcription factors that diverged so long ago that we can't even build a proper phylogenetic tree for these. And so we can look at some of these related proteins and see how changing the ligand and changing the sequence and maybe changing the mechanism of ligand response changes the conformational changes that we can observe by this experiment. We can also look at proteins that have the same fold but are not transcription factors and proteins that are in different oligomerization states to be functional. We're also interested in using structural water molecules as part of a design strategy to predictively design protein allostery and designing proteins that can respond to signals in like a non-monotonically increasing or decreasing way, like band stop filters or band pass filters. There is some evidence that this is possible, but it's a really difficult thing to design and also kind of a rare thing to observe in natural protein systems. You typically need like more than one protein to get this kind of behavior, like a gene circuit, which of course increases the time scale of the response. We're also interested in using the HDXMS data to build new kinds of sensors. And so these projects kind of follow this approach of like you perturb the protein and see what it does to the structure of the protein using an assay, either computationally or experimentally or both, and then use the information to re-engineer the protein to get a different protein function. Okay, so some of the systems that we're interested in are shown here. So this is the LAC repressor. There's also, GPCRs, really good drug targets and fascinating proteins, but these are obligate heterodimers. You can see that this venous flytrap fold is conserved here. But it's a heterodimer that can bind to a lot of different ligands. And instead of being connected to a DNA binding domain, it's connected to a transmembrane domain, which can also bind ligands, in this case, cholesterol, and then is connected to a cytoplasmic side G protein interacting sequence. And so also on the other side, you can see periplasmic binding protein. So these are in E. coli and other bacteria, but they are in a very different cellular environment. They're in the periplasmic part of they're in the periplasm, which is different redox environment, and they can respond to a wide variety of signals, but they are monomeric. And so this guy is connected to a DNA binding domain. This is connected to a cytoplasmic and transmembrane domain. This doesn't have an additional domain. How do these proteins talk to the other domains? These bind diverse ligands. These are dimeric, so they can have a cooperative or ultra-sensitive behavior. Maybe there is some trade-off between binding these ligands and getting this kind of cooperative response. And so it'd be nice to be able to take any ligand you like and get it to bind particular scaffold from the LAC-I-GalR family because they have these desirable features of cooperativity that can be tuned, and then connect them to functional domains of our choice, not necessarily DNA binding domains, right? And just decide like, okay, I want this to turn off or turn on in response to binding my signal, or binding something else on the cytoplasmic side of the protein, for example. So there's a lot of handles to engineer here, right? Like, what ligand do they bind? What kind of cooperative response can they have? And what kind of functional response can they have? And how does it work? So Kyle Weber, a student in my group, is working on redesigning these ligand binding sites to make new kinds of transcription factors for biological sensors. And so far, we're focusing on ligands that are like plastic degradation products to do like continual microbial processing of plastics, and also ligands that are found at high concentrations in solid tumor microenvironments towards like imaging and immunotherapy applications. And so this is adenosine. It's you find it at order of magnitude higher concentrations in the tumor microenvironment. And what we're doing is adopting a protocol developed in the Cortemo lab by my lab mate while I was there called binding sites from fragments, but we're adapting it to work in our scaffolds. So the way it works is we take adenosine or another small molecule, break it up into smaller fragments, and then mine the PDB for particular interactions with proteins, and then try to reconstitute combinations of these hits into the geometry of our scaffolds. So we're using lac repressor, and as you can imagine, purine repressor is a good candidate for binding this ligand, and then screening for good responses. We're doing that on the surface of yeast using yeast surface display. We have a couple of preliminary hits, but the way this works is that you make your computationally designed library attached by Aga2P, standard yeast display system, and then introduce a biotinylated DNA, and then can do the experiment in the presence and absence of your ligand to look for differential response by adding a strep tagged fluorophore and doing flow cytometry. And so when we do this experiment, we're looking for things that that can only bind DNA in the presence of the target molecule, or can only bind DNA in the absence of the target molecule. But of course, this is like just a design of the ligand binding site. What about all of the parts of the protein that we discovered from our HDX-MS experiment that are important for allostery, but are not in the ligand binding site? And so I think to really predictively engineer these, we kind of need to see what's important in this protein fold for the signal transduction. And so the way we can do that is by trying to deconvolute the role of ligand binding from the role of oligomerization in the scaffold. And the way we can do that is maybe to look at other proteins in this family of transcription factors. And so Malcolm, my graduate student, is looking at a variety of LAC-I-Gal-R transcription factors and periplasmic binding proteins, starting with the ribose repressor. And so both of these proteins, they look very similar. They have less than 1.5 all atom, less than 1.5 angstrom all atom RMSD when you compare them to each other in their ligand bound state in pi mol. You can overlay them almost exactly, but they have less than 25% sequence identity. So on the sequence level, they're quite different. They bind different operator sequences, and they bind different ligands. So lac repressor binds to IPTG or allolactose or TMG or ONPF, but RBSR binds to ribose. And so, we can compare RBSR to the lac repressor to figure out what in this fold is unique to being a transcription factor from this fold family. Then we can compare it to ribose binding protein, which also has less than 25% sequence identity to RBSR, but is a ribose binding periplasmic binding protein. So it's like monomeric, has the same fold, and also looks extremely similar. I'm going to show it on the next slide to RBSR. So hopefully by comparing the monomer and the dimer protein from E. coli that look very similar, but both bind ribose, to two transcriptions that bind different ligands, we might be able to do this deconvolution. And we also are including in this experiment a mutant of RBSR that is truncated. So it doesn't have a DNA binding domain as like kind of an artificial intermediate step between looking at the ribose repressor and looking at ribose binding protein, in case the DNA binding domain here has like a strong effect on the HDX-MS for this protein. And so we have some data here that I can share with you. So when we have the ribose binding proteins, we can compare their HDX-MS to see how do these proteins bind to ribose, and how does the oligomerization state perhaps contribute to these effects. So RBSB ribose binding protein looks like this. I'm showing here residues colored in blue that are rigidified in the ribose bound state as compared to the apo state. Mostly we get rigidification when we add the ribose, which makes sense because it is binding. When we compare colored in the same way one of the protomers RBSR to RBSB, we can see a different pattern of rigidification in the repressor as compared to the ribose binding protein. So some examples of this are shown here. So we get this loop seems to be like strongly rigidified when bound to ribose in the periplasmic binding protein that is monomeric as compared to the repressor, which is dimeric. And so here I'm showing a couple of plots where on top you can see the uptake plot for RBSB, increased exchange in the apo state as compared to the ribose bound state. And here, I'm sorry that the ribose bound state is blue instead of red, but all of these states kind of behave the same, and there's a little bit of a difference, but not too much. On the other hand, and I'm showing a different protomer color here just because it's easier to see for this peptide. For this peptide, we see increased rigidification in response to ribose binding in RBSR, but nothing going on in RBSB. And so the uptake plots are here, where almost the same amount of exchange in our periplasmic binding protein, but a stark difference in the ribose bound state in blue and the apo state in black for the ribose repressor. And so we think that these two proteins bind to ribose quite differently as a result of being in different oligomerization states. This is something that we're following up on by looking at this no DNA binding domain version of the protein, and also by doing ribose titrations and DNA titrations in this experiment. But it kind of begs the question, like, how did this ribose binding behavior evolve in the first place? You would think that so let's say you're a bacteria, right? And you see a new sugar in your environment, you want to make a transcription factor to bind to that sugar to do regulation of gene expression in response to the sugar. Do you take a protein that you already make that binds to that sugar and add a DNA binding domain to it? Or do you take an existing transcription factor and just remodel the ligand binding site to bind to the new sugar? At least in this case, we think that this and some bioinformatic data point to like the second scenario, but like interesting thought experiment that we can't really prove. What about in the ribose repressor, the effect of ribose binding versus DNA binding? And so when we have ribose binding in the apo state compared to the ribose bound state, again, the regions colored in blue are rigidified in the ribose bound state. We can see like particular patterns of rigidification here, and part of the protein is derigidified in the ribose bound state as compared to the DNA bound state. And we see a little bit of that here in the DNA binding domain as well. When it binds to DNA, really the whole N-terminal region of the protein becomes rigidified. When we do the ternary complex of both ribose and DNA bound to the protein, we see this like intermediate effect. And this changes depending on how much DNA and ribose we bind, obviously, in the experiment. When we compare the DNA bound state to the ribose bound state, we get this picture, which you can think about in comparison to the lac repressor experiment, where we're comparing the IPTG bound state to the DNA bound state. There is one helix that is extremely similar in these two proteins for the inducer bound state as compared to the DNA bound state, which we think is really cool. So I'm going to show this helix again, right? So in the ribose repressor, we see rigidification at the N-terminal part of this helix and derigidification at the C-terminal part of this helix in the ribose bound state as compared to the DNA bound state, which is very similar to what we see in the same helix for the lac repressor. And so we wonder if this is like an important helix in transitioning between these two functional states in this family of transcription factors that might be independent of the actual ligand that is involved in the regulation. And so one of the applications that we're chasing, now that we know a little bit more about how these transcription factors work, is trying to build them into artificial cell receptors. And so I talked a little bit about how it would be great to be able to predictively engineer GPCRs, but what if we just take these transcription factors and turn them into our own cell receptors? So what we're doing, for starters, is just taking natural LAC-I-Gal-R transcription factors and turning them upside down on the cell membrane, adding a transmembrane domain, and attaching them to split reporter systems like this. So we have two different repressors here that each respond to a different ligand, and they're connected to a split nanoluciferase system, so that, you know, how split nanoluciferase works when the two parts come together and complement. Due to the increased local concentration, you get like complementation, and then you get a intracellular luminescent response. We're doing this in HEK cells, human cells, right? So then when we add this chimeric operator, where we have both the LAC operator and the ribose operator, then we get like clustering in the cell membrane from these two receptors both binding to the DNA, because they still have their DNA binding domain. And then when we add our different ligands, still wild type ligands here, because Kyle's project doesn't work yet, but we can see that they release the DNA just like they do in E. coli, right? So we get them diffusing apart in the membrane when the inducer molecules are added to cell culture. And we can see this on the extracellular side of the membrane from flow cytometry experiments, where here in the red traces, we're looking at how much of a strep fluorophore we can observe in a flow cytometry experiment. So if we see a signal here, it's because DNA is still bound to the cells, even after we washed off any unbound DNA in the experiment. And so we see an increase in the signal when we have DNA and one of these transcription factors in the experiment, but when we add these inducer molecules, we see a decrease in the signal again. We can also see a response on the intracellular side from observing the nanoluciferase signal. So when we add the DNA to the cells, we see an increase in the luminescent response. When we add both of the inducer molecules, we see that the intracellular luminescence goes down. These are controls on the right here showing what happens if we don't have one of the transcription factor receptors, and what happens if we have a positive control in the cell, which is full-length nanoluciferase. We can also see this is pretty modular. So in our system, if we take out the promoter and put in like a lower copy, how do you say, like lower strength promoter, like ubiquitin promoter, and replace the split nanoluciferase system with a dimerization dependent fluorescent protein system, we see the same kind of effect. So now we're working on increasing the dynamic range of these artificial receptors and trying to get them to respond to non-native molecules using protein design, like adenosine, which we hope to be able to repurpose down the line for like tissue building or imaging in real time of tumors, or even next generation CAR T technologies. So I will end here. Thank you for listening to my talk. I will like to acknowledge my lab. We are very young, so we have existed for almost 1 year. I talked a little bit about Malcolm's work and Kyle's work. I did the Black repressor project in Tanya Kurtena's group at UCSF. This is Tanya, and in collaboration with Susan and Helen at UC Berkeley. And Zion was our summer intern who did a lot of the Rosetta work with the water molecules. And Sita actually like binding project that we're working on now as a rotation project when I was a postdoc. So happy to take any kind of questions.
Wonderful, thank you. So if you have any questions, please raise your hands or write it in the chat. Let's see, maybe I'll ask a question to begin with. So so that was really neat how the inducer and the anti-inducer lead to differential effects on the stability of a single helix that then affects DNA binding, right? That's the basic model. So do you see that also in, let's say, B factors in the inducer and anti-inducer bound structures, and does that correlate with the HDX rigidity, etc.?
Yeah, it's a good question. I think if the structures were maybe like higher resolution structures, maybe we would see that, but we don't. We don't see anything like that. So most of the X-ray structures that are available for the lac repressor are on the order of like 3 angstroms resolution. For most of them, there are parts of the structure that can't really be resolved. And so maybe just like the data quality isn't there. We did a more in-depth analysis on the water molecules because it would be great to see that kind of trend. But for the most part, we don't.
I see. Cool. Yeah. And then my other question was, have you or has anyone looked at NMR? I mean, my neighbor here until a few years ago was Babis Kalodimos, who really looked at transcription factor dynamics without structure change as one of the determining sort of features of the system. So has anyone looked at LAC-I? I mean, sounds like it should be the first system to look at, right?
Yes. So I've been actually rereading a lot of these older seminal papers recently because we're about to maybe do a little bit of NMR. So the short answer is not really, because like, so a lot of the NMR experiments that were done were just on the DNA binding domain because the protein is kind of big and it's a dimer and it binds to DNA and it binds to ligands. And so what you can do is chop off the whole core domain and just look at the DNA binding domain and do like solution NMR, and the data for that is really beautiful and you can learn a lot about the structure of the protein. The problem is you can't learn anything about the allosteric because the whole regulatory part of the protein is gone. And so I don't know, like, I don't know how to do an NMR experiment. I would really love to do this with the full length protein, and we might try it because I have a colleague here who specializes in like protein nucleic acid NMR structures and dynamics. But like the other thing I was thinking maybe we could do, if the protein is still too big, maybe we can do some like enzymatic conjugation tricks, like just express like the core domain and the DNA binding domain separately. It's a pretty robust protein. I mean, I bet they would both express separately, and then maybe we can like sortase conjugate them together or something, right? So you express one in Silac media, you express the other in regular media, you conjugate them together, you purify only the correct, you just do like an SEC column or whatever, and then you NMR that, and then maybe you can assign the coordinates anyways, in the different functional states of the protein. Something like that would be super cool to do.
Yeah, I think that's certainly possible. What Babis did and what many people do in that field of looking at large proteins is that you can only differentially label certain amino acids with some kind of isotope. So like leucine, isoleucine, valine, and then you can do some sophisticated NMR experiments with lots of alphabets in their names to essentially look at the dynamics of these methyl group rotations and things like that. And that often gives you, how the dynamics are changing in different parts of the structure. And then the signals become easier to operate. But yeah, I think that's also a neat idea, what you're suggesting, to differentially label things. And that way, you might be even able to get the whole structural ensemble, right? Based on maybe NOEs or things like that. So yeah, cool.
Yeah,
very
I would
exciting.
love to try it.
Yeah.
I mean, I wonder if just like NMR, like maybe it's a thing that now you can do by NMR. It's not too big a system, whereas like 20 years ago, it wasn't possible. You can just do like a straight up NMR experiment now that you couldn't do previously. I'm not sure.
Yeah, how big is the protein?
It's 37.5 kilodaltons, but it's a dimer, so closer to 80. The DNA is pretty big.
Yeah. Unfortunately, the tumbling properties of proteins haven't changed in 20 years. So it seems like that's the bigger thing. But, I mean, they maybe with a higher field or something, it may be easier now. You're right.
Just make the magnet bigger.
Right, exactly. Now they have 1.1 gigahertz or something like that. But anyway, yeah, let's see. Questions? Sorry, I'm just trying to keep track of. Okay, well, I guess maybe the students will be able to ask questions in the session right away. So let's thank Anum for a wonderful talk. Thank you, and we'll see you in that other session. You have the link for that, or Anum? Or?
Yeah, I have it.
Okay,
yeah.
great.